Hello, BISC130. This is the beginning of recorded lecture 1.5. We're still in chapter 4, cell structure. We had briefly started this chapter in the previous recorded lecture, and in that one, we talked about cells in general and some of the structures that all cells have, prokaryotic and eukaryotic cells. But now we're going to start talking about the more complicated structures possessed by eukaryotic cells. So in the notes in the previous lecture, I had written that eukaryotic cells have membrane-bound organelles. So for the next while, we're going to talk about these membrane-bound organelles. So, double underline this because there are going to be several things underneath this. So first and foremost, and if you remember the key terms definition of a eukaryotic cell way back from chapter 1, part of the definition is that a eukaryotic cell has a nucleus and a prokaryotic cell does not. So in some ways, the nucleus is the most important membrane-bound organelle that eukaryotes have. I'll be returning to this figure over and over again, sort of showing plants v animals. And yeah, the nucleus is obvious in both of these. In most cells, the nucleus is the most prominent organelle. It's the biggest, most central thing. Some plants have a water storage thing that gets to be pretty large, but in most animal cells, it's the biggest, most obvious thing. In fact, here are some fluorescently stained, I believe these are fibroblast cells, animal cells, and yeah, the outline of the cell is stained in green, so you can see, the total size of the entire cell, and then the DNA, the nucleus, is stained in blue, and yeah, this is dominating about, a third of the entire cell's body volume. This is a huge organelle. So what does it do? Well, the nucleus stores genetic information. Remember, all cells have genetic information, have DNA, but the job of the nucleus is to store this, to keep this protected, to keep this away from all the other junk in the cytoplasm, an organelle dedicated for this storage. And yeah, like I said, typically the most prominent organelle. If we zoom in on the nucleus and look just at this and not the entire cell, we could see, oh man, there's a lot of stuff labeled here, but there are only a few things that I really want to point out as far as nucleus anatomy goes. One of these is the fact that the contents of the nucleus are kept separate from the rest of the cell, from the cytoplasm, by not one, but two lipid bilayers. Again, really important stuff inside. So yeah, there's inner membrane, outer membrane. It's two bilayers that's separated. This structure as a whole is referred to as the nuclear envelope. Again, just drawn as like a line here when we zoom out really far. But yeah, two bilayers here, nuclear envelope. We do need to be able to go in and send stuff out. This is done through structures called nuclear pores, which very carefully control what gets to go in, what gets to go out. Again, highly protected part of the cell. So nucleus is bounded by the nuclear envelope. And again, we just saw that. The key terms define this as a double membrane structure that constitutes the nucleus's outermost portion. So yeah, the nucleus is membrane, double membrane. And yeah, nuclear pores control entry and exit. What else we got here? Well, this is such a large organelle that it has its own cytoplasm within this structure. The cytoplasm of the nucleus is called the nucleoplasm. That's an easy term for you there. Another structure, labeled over here. These are just different cartoons of the nucleus from different textbooks, different resources. This one's a little more 3D, this one's a little cleaner, whichever one looks best to you. But yeah, nucleoplasm labeled here, just the cytoplasm labeled here. The other thing kind of looks like the pupil or an eyeball inside of this thing is a structure called the nucleolus. Again, very carefully, differently spelled, not the nucleus. This is the nucleolus. This is where ribosomes are created. We talked about ribosomes last time, earlier in this chapter. Ribosomes were things that all cells, prokaryote and eukaryote, possess. This is where they are constructed, within the nucleolus. So nucleoplasm, the nucleus is cytoplasm, and the nucleolus where ribosomes are created. Another thing I want to talk about while we're talking about the nucleus is the DNA itself. So we looked at DNA in the previous chapter as a double helix. Once again, two figures from different resources just showing the same thing. We saw DNA as a double helix structure. Well, eukaryotes have what's the technical term? A buttload of DNA. A lot of information, a ton of instructions. In order for all of this DNA to fit within a eukaryotic nucleus, within a eukaryotic cell, it has to be packaged. It has to be wound up. It has to be coiled around these proteins that are called histones. And these protein coils, these histones, wrap around one another and then coil around again. And you get these highly packaged structures referred to as chromatin. So just the terminology here, DNA is packed within the nucleus as what's called chromatin. The key terms define chromatin as a protein DNA complex that serves as the chromosome's building material. So again, this is DNA with histone protein, a protein DNA complex. And again, this is in the nucleus. This is a eukaryote only thing. Prokaryotes have DNA, but prokaryotes don't do this. They don't have chromatin. Okay, so that was a lot about the nucleus, but again, it's a very important organelle. The next membrane-bound organelle I want to move on to is one called mitochondria, or mitochondrion is singular. We can see this in our overview here. Yep, a mitochondrion, this sort of orange. I don't know why. In every single textbook ever, the mitochondrion is reddish orange. I don't know how this convention came to be, but yeah, the sort of reddish orange little pill is the mitochondrion. We see it in animal cells. We see it in plant cells over here as well. The job, if I could zoom in on a mitochondrion here, the job here is to break down food. So we need to, the cell is taking in food, it's taking in energy, it's taking in molecules, and it needs to break down those things to generate energy. Specifically, we need to break down organic molecules to generate something called ATP. Oh boy, we'll talk more about ATP in several later chapters, but this is the usable energy for the cell. So we're going to break down stored food molecules like sugars, for example, and we're going to use that to generate usable energy. Now, so that's the overall function of mitochondria. What about this structure? Because there was a lot of stuff labeled here. Well, one of the most conspicuous features we see, whether it's a microscope image or a cartoon, are these folds. It's not just a flat oval or pill. We have these folds, and these folds are actually folds of membrane. So just like the nucleus, there's an outer membrane and an inner membrane separating the inside of this organelle from the rest of the cell. These folds within the membrane are called cristae. I don't think I'm going to have that written down, but if you're curious, there's a name for these things. But what's their function? So when we look at metabolism and generating ATP in a later chapter, we will see that a lot of these chemical reactions actually take place in this membrane, embedded within the bilayer of this inner membrane. These folds exist so we could cram in more of these reactions. If this were just a flat pill, we would have, this much space, this much surface area for these chemical reactions, but by having all of these folds, we could cram in way more enzymes, way more chemical reactions that, break down foods and generate ATP. So the purpose of this structure is to increase surface area. So how do I summarize that? Many of the chemical reactions in the mitochondria take place in that inner membrane structure. These folds that exist increase surface area for more reactions. Oh yeah, one more weird thing about these organelles, which you may have noticed on this figure, this says DNA. Wait a second, we just talked about how DNA is stored in the nucleus. That's kind of its whole job. So what gives? Well, I'll give you the short version here because we'll get into the long version Ooh, in BISC132, the second course in this intro biology, if you take that, depends on your major whether you take it or not, but the short version is these mitochondria are thought to have once been free-living bacteria. They got trapped by a eukaryotic cell, and they've co-evolved to be now completely dependent on one another. These things can no longer live on their own. They're not considered living organisms. They're fully dependent on the rest of the cell, but they have a little bit of independence. They have a little bit of their old DNA remaining. If you sequence this stuff, it does look like bacterial DNA. And so, yeah, this is strange. Obviously, most DNA is in the nucleus, but these organelles have a little bit of their own independent DNA and some of their own ribosomes as well. So they could use these instructions and build proteins for themselves. So, yeah, mitochondria have their own DNA and ribosomes. Let's continue talking about breakdown. So mitochondria are mostly breaking down sugars. We also want an organelle that could break down fats or fatty acids or lipids in specific. And there's a reason why we need a specific organelle to do this. But first, let me just say it's called a peroxisome, and these are kind of small organelles. They're unfortunately not pictured in this otherwise pretty comprehensive comparison here. If we want to take a look at these in a different textbook's image of a cell, yeah, they're just little membrane-bound organelles, little circles here throughout the cell. Pretty plain looking, nothing really cool about them, no cristae, no folds. So their job is to break down lipids and fatty acids. Why can't we just do that in the cytoplasm? Why can't we do that in the mitochondria? Why do we need a specific organelle to break down lipids and fatty acids? Well, without getting into the biochemistry of it, it turns out that lipid breakdown, just the chemical reaction of breaking apart a lipid, produces a very harmful byproduct called hydrogen peroxide. You've maybe heard of this before. It's the same stuff that is in a dark brown bottle that you'd pour on a scrape or a cut or something like that to help sterilize the wound. Hydrogen peroxide is nasty stuff. It's dangerous to cells. It is, it's really bad. That's why you clean a wound with it to kill any bacteria that might cause an infection. And guess what? Every time you are breaking down lipids, you are producing this harmful compound. So you don't want this just running rampant inside of the cell. We want an organelle that can contain and neutralize this harmful byproduct as we break down lipids and fatty acids. So yeah, the job of the peroxisome is to contain and break down the hydrogen peroxide byproducts as lipids and fatty acids are broken down. So there's not much to it in the organelle other than this, but yeah, this is where we break down lipids and fatty acids and neutralize the harmful byproduct. To continue talking about breakdown, we have another organelle that's involved with breakdown, something called the lysosome. We can see it pictured here, lysosome. Yep, within the animal cell. There are lysosomes within plant cells. They're not as common, but they can exist. We'll talk about things that are animal only and plant only as we come to them, but so far we haven't gotten into any of these. And actually, let me scroll back to clear up kind of a misconception with mitochondria. This is involved in breaking down sugars, and a lot of people associate this with animal cells, that yeah, animals eat food, animals take in sugars, so animals need to break this down. Plants don't need to break down sugars, they do photosynthesis. Well, that's not true. Plants do need to be able to break down organic molecules at, nighttime when there is no sun for photosynthesis, or in parts of the plant that don't do photosynthesis, like the roots. Mitochondria are not animal-only structures. Here's mitochondria in the animal. Yeah, there's mitochondria in plants as well. So yeah, we haven't gotten to any of these exclusive ones yet. They both have mitochondria. They both can have peroxisomes. And yeah, even though it's not labeled here in the plant cell, both of them can have lysosomes. So why do we need another breakdown thing? Well, lysosomes are really good at breaking down large, complicated molecules. Things like damaged organelle. So yeah, this is a mitochondria that's all jacked up for whatever reason. You would need to send this to the lysosome to fully just destroy this, break this down to pieces. Here's another example of lysosome function where a cell is taking in large chunks of food from the outside world. These very large pieces can be sent to the lysosome for destruction. The lysosome is filled with it's a low pH environment. So this low pH environment within the lysosome is going to denature proteins and help break down these big structures. So again, we've talked about breakdown with mitochondria, breaking down pretty much sugars, peroxisomes, breaking down lipids and fatty acids. Lysosomes are great at breaking down proteins and really big things. Sort of the all-purpose scrapyard of the cell. So in summary, the lysosome has a low pH environment that denatures proteins. There are digestive enzymes in here that are built to function at low pH that break down proteins, large food particles, other foreign material, and or damaged organelles. So again, all-purpose, big stuff breakdown organelle. Okay, couple other structures to get out of the way here. Vacuoles and vesicles. So the job of these things is to store and transport stuff. So let's take a look at these. There are some vacuoles labeled. Here's a vacuole in an animal cell. Here's a vacuole in a plant cell. Vesicles are not labeled here because they're really small. The vesicles would be teeny tiny little dots. They're not labeled here, but they're both found in plants and in animals. The difference between a vacuole and a vesicle is not that vacuoles store and vesicles transport. Vacuoles can store, vacuoles can kind of transport, vesicles can store, vesicles can transport. The difference between these two structures is one of size. Vacuoles are big, vesicles are, as we kind of saw by the fact that they're not labeled here because they're so small, vesicles are very, very, very small. But either one of these things can store things. Either one of these things can transport things. We'll see them in action later in this chapter and in other chapters. But vacuoles and vesicles store and transport materials. These are not exclusive between plants and animals, but there is a special type of vacuole that is our first exclusive. This large central vacuole. Again, here's a vacuole within an animal cell, usually fairly small. This very large so-called central vacuole is exclusive to plants. The job of this central vacuole is to store water. Plants need water. When we talk about the chemical reactions of photosynthesis in a later chapter, you'll see why they need so much water. They need way more water than animal cells do. And so this central vacuole is a plant exclusive thing. So central vacuole is plant only. Again, if we're just comparing plants and animals, I'm not putting fungi or protists in here in this chapter. If we're comparing plants and animals, central vacuole is plant only. It stores water for photosynthesis. Well, now that we're on the train of plant and animal exclusive things, let's sort of continue with this. Let's talk about another plant exclusive thing, chloroplasts. So this is plant only. So if we look at this, chloroplast, color-coded as plant only. And we see these green little pill things in the plant cell. We are not going to see these green little pill things in the animal cell. If we want to zoom in on a chloroplast, here we go. Okay. Like the mitochondria, this is another structure that has two membranes, an outer membrane and an inner membrane. And like the mitochondria, again, we've got some weird structures on the inside that kind of look like stacked poker chips or stacked, Oreo sandwich cookies. These structures called thylakoids are actually folds of the inner membrane. So this is a lipid bilayer membrane that's folded up and around with water sort of as the cream filling between these cookies of folded membrane. why would this organelle have so many folds within its membrane? Well, it's actually the exact same reason why mitochondria had folds within their inner membrane. Many of the chemical reactions of, I think I just copied and pasted this sentence. Many of the chemical reactions of photosynthesis take place in the inner membrane. These structures, these thylakoids, increase surface area so that you can have more of these chemical reactions. And the chloroplasts do photosynthesis. Unfortunately, I like this figure, but it's not labeling the ribosomes or the DNA, but just like mitochondria, chloroplasts do have their own DNA and their own ribosomes. And the story is the same as it was for mitochondria. Again, it's more of a topic for BISC132, but these chloroplasts were once free-living photosynthetic bacterial cells. They got captured by an ancestral eukaryotic cell. They've evolved this codependent relationship. This is now no longer an independently living organism, but it does have some of its own DNA and ribosomes as sort of a relic of its past and being a free-living thing. So we've talked about a couple of things that are plant only, the chloroplasts and the central vacuole. What about animal-only things? Well, there aren't too many of these if we're comparing plants and animals, but there is an animal-only thing here, something called the centrosome. So we can see this labeled in our big overview. This says centrioles. So why does this say centrioles animal only? Well, this is a centrosome. It consists of two centrioles. So you could call this a centrosome. You could call this the centrioles. Same thing. So this is a structure that is animal only. The centrosome contains two centrioles. These things organize microtubules. That's what these tube-like structures are. Looks like a tube, looks like a straw. They named this good, calling these microtubules. These centrioles serve as an organizational structure for microtubules. What do microtubules do? Well, once again, I'm going to kick the can down the road and say we'll talk about these more in a later chapter. For now, I'm just going to say they're used in cell division. So a thing that confuses students is, well, don't plants need to do cell division too? Like, if this is an organizational structure for cell division stuff, why is this animal only? Well, that is, incidentally, a very good question. Plant cells do cell division, but they just don't use centrosomes to make this happen. So the reason why animals use centrosome and centrioles to help organize their cell division and plants don't isn't well understood. It has to do with some of the other plant cell physiology here. But the important thing is these centrosomes, these specific structures for microtubule organization, are animal-only structures. Okay, let's move on to a big one next. The next one I want to talk about is the endomembrane system. And this is a big one. It's a whole system. This is actually going to have 3 different parts to it. But before we do that, let's take a look at this on our overview. This doesn't even say endomembrane system. It lists the 3 parts. These 3 parts that all kind of look the same. They all kind of look like folded blobs, sort of flattened, elongated blobs of stuff. We see it in a plant cell here. We see it in the animal cell here. This is a very large structure, or technically 3 separate structures that are all part of the same overall system. This is very large, taking up a lot of space within the cell, and you'll notice it's associated with the nucleus. We'll see why in just a minute. So all 3 of the components, again, endomembrane system, 3 components. These are all composed of folded sheets of membrane. That's what these sort of blobs are. We're looking at a cross section. We've cut this thing in half to view it, but these folds here, these structures, these blobs, this is made of membrane, lipid bilayer membrane. They're all made of that stuff. And yeah, as we've noted, they're attached to the nucleus, attached to the nuclear envelope. If we want to zoom in a little bit on these structures, we can look at it here. And again, just two different figures from two different textbooks. If one, looks better to you or whatever, they're both showing basically the same thing. And we could see, this one only shows two of them, but this shows all three, color-coded for our convenience. The 1, 2, 3 components of this endomembrane system. And yeah, they're all made of the same stuff, folded membrane sheets. So, what are these three components? Well, let's start with the one that's closest to the nucleus. This structure is called, the blue one, is called the rough endoplasmic reticulum. It's called rough because, whether it's a cartoon here or whether you're seeing a microscope image, I don't have any on hand to show, but even under a microscope, you can see these little dots, these little studs here on this folded membrane. These studs are ribosomes. Should look familiar in their tiny little red dots here. Remember from last recorded lecture, ribosomes are protein factories. They're structures that are responsible for building proteins. So that leads me into the function of this overall endomembrane system. The big picture function of this whole system is to build proteins, modify proteins, and ship proteins to where they need to go in the cell. The eukaryotic cell, whether it's plant or animal, is a big place. And yes, there are some ribosomes that you could see just floating around in the cytoplasm. Some proteins are just going to get made and then float in the cytoplasm as well. But again, it's a big place. We really need something that is going to package and distribute proteins to all the various different organelles and pieces and locations of the cell. That's what this endomembrane system is doing. Shipping, packaging, transport, and modification. A lot of proteins can't just, get built out of amino acids. They need to be tinkered with in various ways, and we'll see that as we move through this system. So, starting things off, endomembrane system. I said there were three components, so let's number them. Number 1, the rough endoplasmic reticulum. Endoplasmic reticulum is quite a mouthful. It's usually just abbreviated as ER, so this is the rough ER. It's studded with ribosomes. Proteins are made here and then inserted into this system. There are some modifications that are done to proteins at this point in the rough ER, but most of our modifications are going to be done later within this overall system. So, okay, here's our rough ER. We've got proteins, we've got a little bit of modification. What about the next step? The next step, which again, not labeled in this one, but it's labeled green in this figure, is called the smooth endoplasmic reticulum. It is smooth because it is not studded with ribosomes. Pretty easy to remember which one has ribosomes and which one does not, which tells you right off the bat, this isn't going to have anything to do with proteins. There are no proteins being made in the smooth endoplasmic reticulum, obviously, because there are no ribosomes. So what is happening here? Well, in the smooth ER, we're making carbs and lipids. We're making the other stuff. So smooth ER, number 2, no ribosomes here. That's why it's smooth. Carbohydrates and lipids are made in the smooth ER. This is important because carbohydrates and lipids are going to be involved in the third step. Some of these modifications that are going to be made in our third component involve carbohydrates and lipids. So that's why part of this sort of pipeline of things involves this structure building carbs and lipids. Now, this doesn't fit the overall narrative of the endomembrane system being, a package and distribution and modification system or whatever. But, sometimes organelles do multiple things. The smooth ER, yes, it builds carbs and lipids that are going to be used later in this pipeline. The smooth ER also breaks down some foreign compounds, things that are complicated to deal with, not just your normal sugars, which would be broken down by the mitochondria, not just your normal lipids, which would be broken down by the lysosome, not your normal Oh, yeah, peroxisomes, not here. Not your normal lipids by the peroxisomes, not your normal proteins by the lysosomes, not your normal sugars for the mitochondria. Sorry if I said that a little wrong a second ago, but you remember all this stuff. By foreign compounds, I mean really tricky kind of things that can't be broken down by any of these organelles. The smooth ER breaks down some of this stuff. Now, we've made proteins in the rough ER. We've made carbs and lipids in the smooth ER. The products of these two things are going to be sent via vesicles. So yeah, I said we'd see vesicles again. And actually, if we go back to this, yeah, look at this. Sorry, the text is so small, but yeah, transport vesicles. So we made some proteins here in the rough ER. We're going to ship that along to the third and final component in a vesicle. It's being used to transport here. Proteins and lipids from the ERs are sent via vesicles to our third component, the Golgi apparatus, or the Golgi body. I like calling it a Golgi apparatus, but apparatus is just too fun to say. So here's our third component in orange here, which is going to finish things up here. So yeah, number 3, Golgi apparatus. We get additional protein modifications. So what do I mean by modifications? Well, some proteins will have lipids or carbohydrates attached to them. So that's why it's important that we're sending the products of both of these things to the Golgi apparatus. Proteins get lipids attached. Proteins get carbs attached. Some proteins get cut in half and then pasted back together again in a different way. Again, there are a ton of protein modifications that exist within the cell. This stuff is going down mostly in the Golgi. Then we have to send these to their final destination. We have to take these modified proteins, package them up into, once again, vesicles, our all-purpose sort of shipping container here, and sent them to where they need to go. Even though these are two different textbooks, they're actually using the same example. In this example, our modified proteins are packaged up into a vesicle and shipped out of the cell. Whatever these proteins are, they have some sort of job that they're doing outside of the cell, so that's where they are sent to. But again, you could have vesicles coming off of the Golgi body being sent to the nucleus, being sent to the mitochondria, being sent to the chloroplasts, being sent to any of these other organelles. How are these organelles going to get the things that they need? Well, they're going to receive shipments of vesicles from this overall endomembrane system. So once this gets packaged up, it could go to lots of different places within the cell. It doesn't have to go out. That's just what both of these figures are showing as an example. So packaging, and then just in general, shipment. Important note here, again, this modification seems like a minor thing. There are lots of proteins that will only work properly if they have been modified. So going through this process is very, very, very important. Okay, so far, all these things we've been talking about fell under this category of membrane-bound organelles. The last several things we've talked about all had their own membrane, keeping them separate from the rest of the cell. But there are several other, labeled things here, very important components of eukaryotic cells that are actually not membrane bound. So now we're going to move over to what I'm double underline heading other not membrane-bound structures. So these are still important for eukaryotes, but yeah, they're not membrane-bound. So let's talk about cell wall. We've been talking about internal structures, stuff on the inside. But if we want to talk about structure and stability, structural integrity, strength, support, the cell wall is a structure that's going to support these cells. Importantly, the cell wall is plant only if we're talking about plants versus animals. Animals, we're gonna have our own way to support the shape of the cell, but having a thick, rigid wall is something that plants do and animals don't. Although, interestingly, I can go back to this, prokaryotes actually have a cell wall. Bacteria actually have a cell wall. So I don't want to call this plant only. I want to call the cell wall not in animals. So prokaryotes have a cell wall, plants have a cell wall, animals do not have a cell wall. So here's how I'm going to phrase that. Cell wall is rigid external protection and support. Made of polysaccharides. We talked about cellulose in an earlier chapter. There are lots of different polysaccharides that could be used to give strength to a cell wall. And yeah, I'm using this phrase, not in animal cells. So yeah, here's zooming in specifically on the plant cell wall. This is here's the plasma membrane, relatively thin structure. Here's the plant cell wall, very thick, very rigid, very protective. And yeah, here's a copied paste slide from a chapter ago. Here's our cellulose polysaccharide being involved in building the plant cell wall. So not much to it other than that, big old polysaccharide, strength and support. But what about animal cells? So we don't have a cell wall, but we do have something. We do need a way to give strength and support to the cell. Instead of a cell wall, animal cells are going to use something called the extracellular matrix. Unfortunately, it's not pictured in this big old overview here, but if we zoom in on an animal cell, we can see the extracellular matrix. And this is it. So here's our plasma membrane. The extracellular matrix is not a thick, rigid wall of polysaccharides. The extracellular matrix, it's still outside of the cell, but it is a loosely packed amalgamation of carbohydrates and proteins. So this is strong, but it is a lot more flexible than this cell wall, which makes sense. Plant cells are, rigid and held in place. They're not really running around or needing to squish, but, animal cells need to be a lot more flexible. They need to be able to change their shape a little bit. They need to squish around as you move. And so having this external protection that is strong but flexible just makes sense for animal cells. So this extracellular matrix, or ECM, if we need another abbreviation, is flexible external protection and support. It's made of proteins and carbohydrates, and yeah, this one's animal only. Plants don't have an ECM, bacteria or prokaryotes do not have an ECM. So let's continue talking about other non-membrane bound structures and talk about something called the cytoskeleton. So again, I like this figure, but it doesn't show absolutely everything. The cytoskeleton is not pictured in this overview, but we can see it here. So the cytoskeleton is, it's all this stuff in between the organelles. So most textbook figures show a lot of empty space. It looks like there's a lot of empty space in between this vacuole and this mitochondrion, and in between this mitochondrion and the rough endoplasmic reticulum. But in reality, there's not a lot of empty space. These fibers, these cytoskeleton structures, some that are drawn orange here, some that are drawn green here, they connect everything to everything else. So everything in the cytoplasm is connected to everything beside it through this complex system called the cytoskeleton. So it connects everything within the cytoplasm of a cell, and there are three major types of fibers. So once again, we'll number these and count to three. The three different components of the cytoskeleton include, number one, microfilaments, also known as actin filaments. So we can see them here. It's a big old string of beads here. Each one of these individual blobs is a protein called actin. That's why the overall structure is called an actin filament. It's just a bunch of actin protein all strung together. So and you can see it labeled up here, actin filaments. They're the red ones down here. Now, what do these actin filaments actually do? Well, I want to take a step back and come to this word cytoskeleton. So it's called the cytoskeleton because it's inside of the cell, kind of like our skeleton is inside of our body, providing structure and support on the inside. The analogy to our skeleton breaks down a little bit because our skeleton is set in its shape. Our bones are not changing their shape throughout our life. They're going to move against one another at our joints, at these points of articulation, but our humerus is not going to dissolve and reform. Our skeleton is pretty set. However, the cytoskeleton is not set in place. The cytoskeleton is something that can dissolve and rearrange dynamically. I pulled this from an immunology figure, so immunology stuff really gets into alphabet soup. Do not sweat any of this stuff. But my point here is this figure is showing how actin can be constructed. These filaments can be constructed in order to dynamically change the shape of the cell. This is from an immunology example where the cell was actually reaching out, changing its shape, grabbing a bacterial cell and eating it so it could destroy it. That's how your immune system works with things like this. It's going to eat stuff and destroy it. So yeah, actin filaments, these are dynamic structures. You may remember this from earlier in this chapter. I said that the entire cell was stained with green. It was actually actin that was stained with green here. Again, these filaments, they're all throughout the entire cell. So if you want to see the entire cell, you could stain actin and see the entire cell. And the third little thing here is, actin. This is a figure that I've stolen from the BISC132 chapter on the musculoskeletal system. This is illustrating how muscles work, how muscle contraction works. And yeah, it's going to involve this protein actin. It's going to involve these actin filaments. So they're involved in several different things. So how do I summarize this? Well, microfilaments, also known as actin filaments, can disassemble and reform to let the cell change its shape. Again, a dynamic structure. And again, not getting into the details in this class, but you should know that actin filaments are involved in muscle contraction. All right, that was one type of fiber. I said there were three. Next, let's move on to our second one called intermediate filaments. It's kind of a weird name, intermediate. They're called intermediate filaments because they are intermediate in size. They're a little bit thicker than actin filaments, but they're a little bit thinner than the third one we're going to talk about. It's a dumb way to name something, but it is what it is. So what do these intermediate filaments do? Well, their whole job is not nearly as complicated as actin. The whole job of intermediate filaments is to be strong, to give strength to cells. Keratin is an example of a filament protein. Our hair, our fingernails, if we want to look outside of humans, the horn of a rhinoceros, for example, these things get their strength by being filled with intermediate filaments. So yeah, their job's not that complicated. It's just to be strong. Intermediate filaments provide structural stability. Now there's a third one. Our third component of the cytoskeleton is something that we've actually seen before, microtubules. So if we want to look at them here, yes, in green, once again, labeled up here, microtubules. Yep, it is a tube. Yeah, we saw these before when we were talking about centrosomes and centrioles. The job of microtubules is actually twofold, two very different jobs. So what I had said before, when we were talking about the centrioles and the centrosome, I said that microtubules were involved in cell division. And, that's still true here. When we get to our chapter on cell division, we're going to see these, I don't know why these conventions, every textbook does it the same way. I guess it's convenient. I don't know how they decided on this. These green, always green for some reason, microtubules are going to play an important role in cell division. But also, these microtubules play a very important role in transportation. So if I can back up to when we were talking about the endomembrane system, and we saw these vesicles being shipped from the rough ER to the Golgi, and from the Golgi to somewhere else in the cell, these figures kind of show these vesicles just magically floating to where they need to go. Well, they're drawn this way for simplicity, but nothing is magically going to float where it needs to go. If you want to ship proteins from one part of the cell to another part of the cell, you're going to need a highway system or a railway system or whatever your analogy of choice is for transportation. And that's what these microtubules do. Here's a vesicle packaged up with whatever being shipped to wherever. Here are some very cool motor proteins that are going to make this thing zip along, and the microtubules are serving as a track for these motor proteins. So yeah, two very different jobs by the same cytoskeletal fiber, playing a role in cell division and being a highway system for vesicles to move around. So how do I summarize this? Well, microtubules, they are hollow tubes. Yes, a little bit hollow on the inside. Key role in cell division, and these microtubules play an important role as transport rails for vesicles. Two very different jobs, but done by the same cytoskeletal fiber. Okay, we're almost done. Just a couple more of these other not membrane-bound structures, flagella and cilia. So first off, I want to say these are optional structures. So our big overview, it's not showing flagella on either of these cells. It's not showing cilia on either of these cells. And, that's fine because, again, these are optional structures. Some cells will have flagella, but not cilia. Some cells will have cilia, but not flagella. Some cells will have both, and some cells will have neither. So what are these things? Well, these are structures that exist on the outside of the cell. Flagella are these long whip-like structures. Cilia are these short, sort of fuzzy hair-looking structures. Most cells that have flagella have only one or maybe two. Cells that have cilia typically have lots of cilia. So that's what they look like. Again, flagella, long, cilia, short. What do they actually do? Well, flagella are involved in movement. Again, both of these are defined in the key terms. Sorry, my rattling of paper here. Flagella are defined in the key terms as a long, hair-like structure that extends from the plasma membrane and moves the cell. So yeah, these are used for motility. These cells will whip these flagella around in order to swim through the liquid, in order to swim around. The key terms define cilia as short, hair-like structures. They're both described as hair-like, but I guess you can have short hair or long hair. So flagella are long and hair-like. Cilia are short and hair-like. Short hair-like structures that extend from the plasma membrane in large numbers and function to move an entire cell or to move substances along the cell's outer surface. So you can also use cilia to swim along, or you can use cilia to move stuff around. We're going to see that in the human body, again in BISC132. Sorry, I keep referencing that course, but it comes up a lot in this particular lecture. So moving stuff along. Okay, that takes us to the end of all of these organelles, all of these non-membrane bound structures. If you want, here are a couple of slides with a summary of what we have talked about so far. This is no new information. Anything here, plasma membrane, cytoplasm, nucleolus, nucleus. We've talked about all this stuff already. Oops. It continues into a second table here. This is a summary of everything we've talked about in this chapter so far. It's breaking down whether it's present in animals or plants. Again, this seems like an enormous amount of information, but truly almost everything is found in both plants and animals. It's only the couple of weird things like the centrosomes and the chloroplasts in the cell wall that are plant but not animal, or animal but not plant. So yeah, it seems like a lot to memorize, but really, you just have to remember the things that are animal only, the few things that are plant only, and the few things that prokaryotes have. Because all of these complicated membrane-bound organelles, like a Golgi, like ER, like chloroplasts, like the nucleus, like any of these other membrane-bound organelles, that's a big no for all these prokaryotes. So you could draw this out yourself, you could try to memorize this or whatever, but it's not as intimidating as it seems. And again, these tables are no new information. These are just a good summary of what we've talked about so far. And again, all my slides are available on Canvas if you want to download these yourself, or you could pause now and screen capture, however you want. But again, no new information, just convenient summaries of what we talked about so far. So yes, that takes us to the end of this chapter on cell structures, and conveniently, this is the end of lecture 1.5, and even more conveniently, this is our cutoff for exam number 1. So exam number 1 will have all material from the beginning up through what we've talked about here at the very end, flagella and cilia. This is the end of 1.5. This is a cutoff for exam number 1.